Hi, everyone. I'm so happy to be here. As scientists, we get very few opportunities to engage with the public like yourself, and so I'm very grateful for this opportunity. And I'd like to tell you a little bit about what our lab has done and what we are interested in doing. Now, all of you have heard of Star Trek, where they jump on a spaceship and they make a voyage to explore outer space. My lab explores inner space. And we are interested in looking at the last bastions, the last spaces and places inside our bodies where we have no idea what is the kind of environment that we will find. And these spaces are spaces that are present inside our organelles. So what are organelles? Inside our bodies, we have a range of different kinds of cells, all of different shapes and sizes. You'll see that the ovum is nearly 30 times the size of a sperm cell. Rod cells and cone cells that are present in our retinas are very different shapes from a dendritic cell, which is a kind of immune cell that we have. But common to all these different kinds of cells, regardless of their size, their shape, their function, is that they have certain small subcompartments inside the cell called organelles. And just like organs of the body perform very specific functions for the body, organelles inside our cells perform very specific functions for cells. Inside the membranous compartment of an organelle, there is a space inside, and this is called the lumen. And inside the lumen, you have very, very different kinds of biochemical reactions, where each organelle is performing a very specific class of biochemical reactions. And these reactions are extremely important for us to stay healthy. And so, one of the things that I was very curious to ask was what is inside, what is the kind of chemical environment that promotes this unique chemistry. For example, an organelle called the mitochondria has a lumen where the chemistry is such that it's mass-producing ATP. There's another organelle called the lysosome, where the chemistry inside is such that it's chopping up your proteins at massive speeds into amino acids. But what is this microenvironment? Are the levels of sodium, potassium, calcium, chloride, acidity, is it same as the cytoplasm or different from the cytoplasm? If it's different, how different is it? And so, curious to answer this question, I developed a technology based on DNA, a nanotechnology that's based on DNA, that goes inside our organelles and measures the levels of these ions inside them. And in doing so, it has allowed us to open up the chemistry and the biology of organelles in new ways. And so if you ask, how are ion levels inside our cytoplasm regulated? We have a plasma membrane, and outside the cell, we have very high sodium but low potassium. Inside the cell, it's the opposite. You have low sodium and high potassium. And 1/3 of all the energy every cell produces goes towards maintaining this difference in ions. And so, because ions are charged, and the plasma membrane is also charged, ions cannot freely pass through the membrane. And so the body has figured out to use certain kinds of ion transporting proteins called ion channels. And when they open, a specific ion channel, let's say sodium ion channel, when it opens, sodium moves from high concentration to low concentration down this pore. And potassium, when a potassium channel opens, it moves from inside to outside. Again, high concentration to low concentration. Why is it important to know about these ion transporting proteins? Because the ion transporting proteins are the single largest class of proteins against which there are FDA approved drugs. these proteins are present on the plasma membrane. And nearly all the drugs that we are taking, that are FDA approved, the largest classes of drugs, are molecules that will force an ion channel to either open or block it, and they change the activity of the ion channel. And so imagine this. This is all looking at proteins that are only present on the plasma membrane. But the plasma membrane is only 2% of total membrane inside the cell. The remaining 98% is present in organelles. And organelles also have ion channels. And we have not been able to look at the activity of organellar ion channels that are present inside cells in their native environment. It was intractable until we developed the technology to look at ions inside organelles. And so now that we have developed this technology, we can suddenly access a vast and unexplored landscape for biology and medicine. And so why is it important to know this? Why is it important to study these ion channels? Because when they go wrong, when there are defects in such organella ion channels, it results in diseases that are cardiovascular, neurodegenerative, autoimmune disease, cancer. And so why should we know about ion levels inside an organelle? Because it's the only reliable readout for the function of an ion channel inside an organelle. And the logic is this, that when an ion channel opens, ion levels will flow from high concentration to low concentration. So let us take your favorite sodium channel that's present in an organelle that's maybe related to disease. If I don't know the level of sodium inside the organelle, when it's opening, is the ion going to flow from inside to outside or from outside to inside? That depends on the concentration of ions inside the organelle. Why should I know whether ions are flowing inside or outside? This is going to dictate, if I am designing a drug for that ion channel, it's going to dictate my therapeutic strategy. Should I be thinking about making a blocker, or should I be thinking about making an opener, right? And so that's why it's really important to know the ion levels inside organelles. And so in order to develop this technology, now we've got the answer. In order to develop this technology, we had to overcome 4 different problems rolled into one. And I'm going to tell you what each of these problems were. The first one is the problem of measurement versus detection. It's a how much of it is there, not an is it there question. And so here I'm going to ask you to participate with me in a small experiment I'm going to conduct on you. On the top panel, you see different panels of different squares where you have different intensities of only one color, green, right? In the bottom panel, you have different panels. Again, you have different intensities of green, but I have overlaid it with a red color, yes? So how many people find it easier to tell the difference in green intensity between 2 adjacent panels? How many people find it easier to tell this for the bottom panel? Hands up. I can't see. All right. And the reason is because your brain is computing 2 inputs. It's looking at the ratio between red and green, and that's telling you how much of green there is, right? And so this concept is called ratiometry. So in order to measure, it is always easier to measure with a reference that is not changing, yes? And the second problem is the problem of access. Organelles are very small. The volume inside is a few attoliters, 10 raised to minus 18 liters. You have to put your sensor inside that volume, which is inside an organelle. That organelle is present inside a cell. That cell is present inside a living body. And it's hard to access these spaces with precision. The third is what I call the black box problem. We have no idea of the ion levels we are going to encounter over there. It can be anywhere between nanomolar to millimolar. That is 6 orders of magnitude difference. And so now, if you're looking at any binding-based sensor, you can only quantify in that linear regime, right? And if that linear regime covers only 2 orders of magnitude, but your space is much larger. And so the answer is, we develop a culture of learning from failure. We say, all right, the first time we try it, it's probably going to fail. But the way we fail will help us understand how to succeed next time. And if the sensor fails to turn on, we tighten the binding. If the sensor is completely saturated, then we loosen the binding. And so in this way, we've been able to look at quantify ions. The last problem that's waiting to kill you is the fact that inside all of these organelles, many of the lumens are acidic. And acidity interferes with every kind of ion-sensing chemistry. And so because of this, the chemistry inside organelles remained unmapped, unexplored, and unused in our understanding of health and disease. And so how did we solve the problem? We solved the problem in step by step. The first way is to solve the measurement problem. And for this, we took ion detection chemistry that glows green, and we used nano devices where you have 3 strands, and each strand has one function. One of the strands carries an ion sensing reporting dye, which glows green when it encounters an ion. The second is a strand that carries a red dye, which is always on, and that gives you ratiometry. So together, the first 2 strands give you a measuring module. The next is you have to put this measuring module inside your target organelle of interest. And so that's where the third strand comes in. It helps target the organelle. So what you're going to see is actually, I'm showing you the plasma membrane, and you'll see nano devices getting into the plasma membrane, right? So what happens here is that we use the knowledge about how a cell feeds itself. Right? A cell takes everything that it needs from the outside, whether it's a nutrient or whether it's a hormone. And so our nano devices basically bind a protein that's on the cell surface. And after it's bound the protein, the plasma membrane bends and it forms a little fat bubble. That fat bubble goes inside the cell. And you can see that now this has entered the cell. Now, depending on the protein that it has bound, this nano device goes and gets deposited inside the lumen of a target organelle. This is very similar to how, if you have the right ticket, you can get onto the right bus at the bus station, and that bus will deposit you in the neighborhood, a specific neighborhood of your city. And so what we do here is that we glue a molecular ticket, a molecular signal, onto our measuring device such that it can bind a protein on the surface of the cell. And in this way, your nano device gets accumulated in your lumen of your target organelle, and that allows you to measure the level of ion in your target organelle. And so this simple and modular system has opened up the chemistry inside organelles. And so with this, I'm going to tell you a couple of new things that we've found. We mapped the level of sodium and potassium inside our organelles for the first time. Now, inside the cytoplasm, sodium and potassium are known to dominate cellular physiology and disease. But how they were distributed inside our organelles was not known. And we found that there were huge differences between sodium levels and potassium levels inside an organelle and on the other side of the membrane. That was great, but beyond this, what did it reveal? It revealed something even more interesting. And that was the fact that organelle membranes seem to be checking off all the boxes that the plasma membrane checks off. For example, the plasma membrane also has sodium and potassium gradients, just like organelle membranes. The plasma membrane also has ion channels, just like the organelle membrane. So we know that ions move across the plasma membrane, and the movement of an ion is basically going to give you a current. That is why plasma membranes of all cells are actually electrically active. This is why if you put your finger into an electrical socket, you get a shock, right? Because your cells are electrical and you are conducting. Therefore, you are conducting. That is why you feel a shock. So do organelle membranes also conduct electricity? But for that, ions have to move across the membrane, right? It's not just enough to have an ion gradient and ion channels. Ions must move. And so what we did was develop a nanoscale DNA nanodevice that acts like a voltmeter that tells you absolute value of ions inside organelles. We sent it to organelles, and we found that organelles had high voltage. And this upended our view of organelles that we had always thought of as inert fat bubbles. Now we found that they had electrical behavior, and their electrochemical identities were also quite different from each other. And so this now made us think even a little more beyond that. So in our brains, electrical signals are transmitted across layers of different circuitry. All of them are at different length scales. And I believe that there is a missing link in this multi-scale circuitry that's in our brain. The circuitry that we have allows us to look at disorders, the circuitry that we have allows us to have control over things like movement and memory. But when they go wrong, how do we restore that circuitry? And I believe it's because of this missing link that we don't know, that it has been hard to restore that circuitry. That missing circuitry, I think, is at the scale of organelles, their electrical behavior. Remember that organelles share the same cytosol, and the cytosol is an electrolyte, with the plasma membrane. So I believe that the electrical properties of the organelle can feed back and shape the electrical behavior of the plasma membrane and the electrical behavior of the whole cell. And so one of the things I'm trying to do is to try to understand this new scale of organelle electrical circuitry, because I think understanding it will give us a better understanding of a single neuron as an electrical unit. And that in turn can help us understand how can we fix broken electrical circuitry in our brain when it comes to fixing disorders of movement and memory. Our brain is not our only electrical organ. We know that our heart is also electrical, right? And even there are other organs in our body where huge amounts of ion transport are absolutely critical for their function. And when it goes wrong, it leads to disease. And so I think that our organelle circuitry is going to be applicable even to such cells and such conditions beyond the brain, because cells even in those kinds of organs are also electrical. And I'm going to end my talk with a small story about an old woman. It's an argument between an old woman and a scientist. And there was this belief that the Earth, the globe, was resting on the back of a turtle. And then the scientist was trying to convince this old woman that this is not the case. And so he says, okay, what is that turtle standing on? So she says, an even bigger turtle. And he says, okay, what is that even bigger turtle standing on? A giant, even bigger turtle. And he said, what is that giant turtle standing on? And she said, I know what you're doing. It's turtles all the way down. Today, the situation is reversed. I, as a scientist, am telling you the final message of my talk, which is we are electricity all the way down. Thank you, and I wouldn't be able to do this work. I'm just here on the part of a team, which is most of us are immigrants. And I think immigrants bring risky ideas. We bring new ways of thinking. I myself am an immigrant in the country that I work. And we have this capacity to take on risky work and enrich the society that we are in. And so I do hope that we will all go out knowing that in order to move the edge of knowledge, We need knowledge and approaches from everywhere in the world. And so thank you so much for your attention.